Hi, class. So we've been talking about classification of life on Earth as part of our investigation into what the nature of life is, so that we can convince ourselves what it is we should be looking for when we start going off planet and trying to find life elsewhere in the cosmos. So we talked a little bit about taxonomy last time. So we're going to talk a little bit more about that, not about how we do taxonomy, but how it's actually used to map out life on Earth today. And then we'll talk a little bit about what the fundamental processes of life are, and we're going to use that to get into the way life propagates next lecture. Okay, so let me go ahead and start a couple slides here. Okay, so, when we look all around us, especially at a scene like this behind us, we see a very broad diversity of life. And when you first learn about life in school, you often classify life into two broad categories, plants and animals. And in fact, when you start studying life, what you discover is that there is a great diversity. And that great diversity of life is usually characterized in something called the tree of life. So this is a fantastic artistic representation of the tree of life. This is by a very famous artist I like very much called Ray Troll. Ray's a great environmental artist, and if you are interested in this kind of style of art that you see here, you should certainly check out some of his books. But what we're going to talk about today is the tree of life, and how we arrived at its structure. Okay, so we'll talk a little bit about the variety of life on Earth. We'll talk about how we divide it up, how we actually go about the taxonomical process that we kind of introduced last time. And then I want to spend just a little bit of time in this segment talking about big stuff versus small stuff. Okay? So the whole diversification of life really began in the 17th century with a guy named Van Leeuwenhoek. Van Leeuwenhoek was a businessman. He was a Dutch draper, and he was interested in microscopy, which was very new at the time. So this was the middle 1600s. So it was kind of 50-ish years after the invention of the telescope. And people had discovered that if you play around with glass and lenses, not only could you make things that could magnify things that were very far away, you could also make devices that could magnify things that were very small. And Leeuwenhoek became very good at this. He developed a method for making very good glass lenses, basically by making whiskers of glass and then melting the ends of the whisker in a candle flame. And so he was able to make these just very beautiful, perfect small glass spheres, which he then used as the lens in the single lens microscope of his own design. And you can see him using one of them there in this illustration. So when he discovered he could do this, he started applying his technique to everything around him. And in particular, he discovered the microbial world. And so as a consequence, he's often called the father of microbiology. So in October of 1676, he was the very first person to discover a single-celled organism. So here on the bottom of this page, I have some of Leeuwenhoek's sketches. On the left, there is a sketch of the cross-section of a tree sapling. So you can see what we understood at that time to be the nature of most macroscopic life, plants, animals, big things that walk around, which is it is comprised of many small cells. Okay, and you can see that clearly there in his sketch. But in 1676, he discovered that he could find in a drop of water individual single-celled organisms that appeared to be living and moving and acting with the world on their own. Okay, so not in contrast with any other duplicate cells. So he wrote only in Dutch, but he called these things dierkens, or if someone speaks Dutch, there's probably a proper Dutch pronunciation of that, which was translated into English as animalcules, okay, which comes from the Latin for tiny animal. And so you can see some of his sketches of these animalcules over there on the bottom right. And some of them will look familiar to those of you who have studied microbiology before, parameciums and euglenias and so forth. Okay, so this caused an instant stir in the Royal Society of London, who he had sent a letter to about his discovery, because the idea of single-celled organisms was in complete conflict with everything they thought about life at that time. They, like you and I, when we were taught in elementary school, had divided life very clearly into two groups: plants and animals, and there wasn't really any room for anything else. They had great skepticism for von Leeuwenhoek's discovery, but they sent someone there to have him show them what he was seeing, and to actually observe him observing to make sure he wasn't fooling himself. But in fact, they very rapidly came to the conclusion that indeed he was correct, and he had discovered a new form of life. Okay. So the beginning of the tree of life was what we're all taught. Initially, there were just these plants and animals. It's the bulk of life that we can see around us whenever we look out in the world. And we used to call these things, well, and we still do call these things kingdoms. There's the plant kingdom and the animal kingdom. And so the introduction of microbes very quickly started to throw some confusion on this issue, because microbes didn't really fit any description that you and I might normally have for animals or for the behaviors of animals or the features of animals, but they behaved and they act of their own volition when you watch them in the microscope. And so, it might have seemed natural that you might call them animals. They are tiny animals, animalcules, but they certainly don't look like any animals that had been seen before. And so scientists very rapidly convinced themselves, well, maybe we should have a new classification: plants, animals, and then these microbes, whatever they might be. In a similar vein, there are similar confusions on the parts of plants. So there are fungi in the world. So the classic example is a mushroom, and they kind of act and look like plants as well, right? They grow on the ground, they don't move around, but they clearly aren't the same thing as plants, right? They don't harvest sunlight. They don't necessarily make seeds and go through fruiting stages and all those things you kind of expect of plants, okay? So fungi also are an example of a classification scheme that doesn't fall into that very simple-minded plants and animals thing that we all learn when we first start learning about biological life. Okay, so this causes a great confusion. And when you're doing taxonomy, if you are kind of making up your own criteria to divide groups of samples that you have into different categories, you can just choose whatever criteria you want. But if you're trying to come up with some global taxonomy, which is what zoologists and biologists and scientists are trying to do, then you want to have good understanding of what the broadest possible categories are. Are there plants? Are there animals? Are there microbes? Are there fungi? Are they related? Are they different? Is there anything else? Okay. So this is a subject that by no means has been resolved. Back in Leeuwenhoek's day, and indeed up into the 20th century, much of this classification was done based on physical characteristics, things that you can see, things that you can observe and look at, okay? But in the modern day, what we do, we tend to now characterize the divisions in the tree of life, in our taxonomical classification, based on the molecular biology of the things we're trying to classify rather than their observational character. Okay, and so it'll be a day or so before we get to the discussion of it, but most of you are aware of the existence of DNA. And so much of the molecular biological classification in modern taxonomical description of the tree of life boils down to looking at the biochemistry of the DNA in the subjects. Okay, so as a consequence then, what we've recently come to, and by recently I mean in the last 20 years or so, is that there are three very broad classifications for life. They're called domains. Okay, and within the domains are the kind of classic things that you learned about: the plant kingdom, the animal kingdom, fungi, microbes, whatever. Okay, but the three broad domains are as follows. They're the so-called eukarya, or for those of you who have taken biology before, these are called eukaryotes. Okay, so these include all the large multicellular organisms like the stuff in the picture behind me, plants, animals, and so forth that we're used to. But what characterizes eukaryotes is that when you look at the cells, okay, the individual small components that the life is made up of, those cells have an enclosed nucleus with a membrane within them. And what we know now from our learning of biochemistry over the last many decades is that the nucleus of the cell is where all of the genetic material, the DNA that we're so interested in terms of the biochemistry, is actually enclosed. Okay, so eukaryotes have an enclosed nucleus with a membrane. Now, the other classification is the bacteria, okay? And so bacteria are a microbial class of creatures. They are prokaryotes, which is they are characterized by cells that don't have an enclosed nucleus. So all of the genetic material inside a bacteria is floating around inside the bacteria. Some of them have other enclosed structures inside the bacteria itself, but the characterization that's being made here is always specifically with regard to the nucleus. Okay. Now, the newest part of the newest domain that's been added to the classification scheme is something called archaea. So archaea are also microbial prokaryotes, and for a long, long time, we just thought they were other kinds of bacteria. They are other kinds of bacteria, and they were originally called exotic bacteria. So just like bacteria, because they're prokaryotes, they're characterized by cells without an enclosed nucleus. But what we've discovered is that when you look at the molecular biology, the biochemistry of bacteria and archaea, they clearly are different in very fundamental and strange ways, okay? So they have different genetic histories if we look backwards in their genetic histories, and that seems to be a very distinguishing feature between the bacteria and the archaea. And in particular, one of the kind of very quick things that you see happening with the creatures that we call archaea is that they tend to exist in very extreme environments, okay? And we're going to talk about what we call extremophiles next lecture. But most of the archaea seem to be in that category. What I would say is that the distinction between bacteria and archaea is still an ongoing area of research. The archaea have only been identified in the last 20 years or so. And so there is still a lot of research going on into what distinguishes them from what we call ordinary bacteria, and still a lot of debate and argument about any given microbial organism. Should we classify it as a bacteria or as an archaea? And part of that classification scheme goes back to what you and I have been talking about for the last couple of lectures, which is when we're doing taxonomy, we look at the characteristics of the organisms that we think are important, and we try and distinguish them and sort them. Do they have these properties? We're going to put them in these piles, and if they don't, we're going to put these in these piles. And as we're learning about what distinguishes these two kind of classes of organisms, those decisions that we make about how to sort them are also evolving. But for the moment, this is the way that we classify all life that we know of on Earth, okay? So this is a figure, there's a form of this figure in your book that shows kind of what the modern tree of life looks like. Okay, so the tree of life is very much a tree. It is branching. So the branching shows the more refined classification of organisms based on characteristics as we go up the tree. So as you go down the tree, the organisms all have similar characteristics, and as you go up the tree, they have different characteristics. This is by no means a complete tree of life. You can take any one of these branches, and it will further branch and further branch and further branch until you get to the very end where you uniquely identify a given species of organism on the planet. Okay? Okay, so you'll notice here that our classic macroscopic creatures that we're all used to, fungi, plants, animals, all live down this eukaryote branch of the tree of life. Okay, so they all have nucleated cells, and they are multicellular and complex, and they all live on that tree. That means everything over on the other two branches, the archaea and the bacteria, are single-celled. Okay? Now, this is another representation of the tree of life that you like, that I like. And there are as many different ways of drawing the tree of life as scientists who sit down to draw them, and it's just all about how much detail and how many of the branches and at what level do you want to show all the different branches. But the reason I show this one in particular is I want to draw your attention to this right here, okay? So this is called the root. Okay, so if you follow all the branches of the tree of life back to their common trunk, okay, to the original trunk, and if you go back to the art image from Ray Troll at the beginning, you'll see that this is the same route that you end up at, is the origin of all life on Earth seems to be a single prokaryotic ancestor from which all life derived. Okay, and there is good molecular biology evidence for that, and our classification schemes and our understanding of natural selection, which we'll talk about next lecture, also seem to suggest that. Okay, so this is kind of the fundamental notion about the tree of life, and it's one of the explanations for why we think the biochemistry of all life on Earth is so similar, is the same. Okay, and we'll talk a little bit more about that when we talk about DNA next lecture. Okay, so as you go down the tree of life, all of these branches become ever more refined, and they get into little tinier branches, and there are gazillions of those if we were to extend this to its full extent. And at the end of each branch, at its terminus, is a single individual species on Earth. Okay, and so as biological catalogers who are trying to document all life on Earth, we have and constantly are doing a census of all the life that we can find on Earth, where we count the total number of species that we can find and distinguish them and figure out what their classification is in this big tree of life scheme. So currently today, there are about 1.4 million known species on Earth. Okay, and in rough division, you can see here how many we know of different species in each of the different kinds of categories you might imagine. We know of lots of insects, we know of lots of plants, we know of lots of animals, and not so much of the microbials by comparison, okay? In terms of our understanding of the differentiation of the molecular biology of all the different species, if we look at the variance we see and we kind of make estimates about how much variance there could be, we have estimated that there could be more than 8.7 million different species on Earth. And we only know right now of 1.4 million species, okay? And so the thing you should take away from that is we have no idea what most of the life on Earth is all about. Okay, so we're still looking, we're still cataloging. Every year we put out new catalogs of all the new species discovered. There's tremendous numbers of new species to be discovered, particularly in the deep unexplored parts of places like the rainforests, certainly in the oceans, and the census of biodiversity is just constantly changing. Okay? Okay, so the last thing I want to talk about in this segment of the lecture is I want to think about microbial life on Earth, because the statement that you will often hear us make, and this calculation here is designed to convince you of that, is that the microbial life is far and away the most common kind of life on Earth. Okay, so this is a form of the calculation you can find in the book on page 171. I encourage you to try and do the calculation I'm outlining here yourselves. Do it on your calculator, write it out. And if you want to see a slightly more complicated version of it, you can certainly follow along the example in the book. Okay, so let's start by talking about humans. So there are something like 6 billion humans on the planet. Okay, and if each of us weighs about 50 kilograms, so some of us weigh more than that, particularly now since we're all locked up during the pandemic, we're getting pretty chonky, but some of us weigh less than that. But on average, let's assume that a person weighs about 50 kilograms. That means the total mass of humans on Earth is something like 300 billion kilograms, 3 times 10 to the 11th kilograms. Okay, so that's going to be our foundational number that we use to compare to the microbes. Okay, so now let's just talk about the microbes in the ocean. Okay, we're not going to talk about all the microbes on Earth. I'll make a comment about them in a minute, but let's just talk about the microbes in the ocean, because that's in principle easy for me to measure and easy for me to do calculations with, because I can make a measurement that I know something about. Okay, so let's start. So the ocean is about 1.4 times 10 to the 21 liters of ocean water. Okay, and if I were to go down to the seashore and take a liter bottle with me and scoop up a liter of that ocean water and then count the number of microbes in the liter, I get about 10 to the 9 microbes per liter. Okay, Now, if I look at any individual microbe, okay, and you and I can't do this with an average kitchen scale, okay, but we have techniques for doing this in the biology lab. We can weigh the microbes, or we can estimate what the microbe's weight should be based on their size and their volume and density and so on. Then each microbe has a weight of about 10 to the minus 15 kilograms, okay? So I take the total number of liters of water in the ocean, I multiply it by the number of microbes per liter, I multiply by the mass per microbe, and I get that the total mass of microbes in the ocean is 1.5 times 10 to the 15 kilograms, which is way more than the mass of humans on the planet Earth, okay? It's about 5,000 times the mass of humans on Earth. So the microbes on the planet, just in the ocean, not even counting all the microbes that live on the land, all the microbes that live on you, all of the microbes in the atmosphere, all of it, just the microbes in the ocean outmass all the humans on Earth, okay? And you might not have guessed that, but even if you didn't want to talk about mass, if I go back to the calculation there, 10 to the 9 microbes in a liter of water. Okay, so that's 1 billion microbes per liter. So if I just get 6 liters of water, less than 2 gallons of water, the microbes in 2 gallons of water outnumber the number of people on Earth. So even if they don't outmass us, or if you want to argue with mass calculation, they certainly outnumber us. Okay, and so this is why when we start talking about looking for life elsewhere in the universe, particularly here in the solar system, our attention is focused mostly on microbes, because the assumption is microbial life is going to be more common, because that's what we see right here on Earth. Okay? Okay, so that is all I'm going to say for this segment of the lecture. Okay, we're going to go back and we'll talk about the processes involved in singular cellular level, which will apply both to multicellular life as well as microbial life. And then we'll use that to start talking about life processes and how we're going to look for that. Okay, so have a good time, and we'll talk to you all soon.